last week we finished a conversation with the talk about start of an eye anatomy, right? We talked about the eyeball being separated into two segments, correct? Oh, sorry about that. I'll switch in a second. Eyeball separated into segments, the anterior segment and the posterior one. And that anterior segment is further separated into two chambers, the anterior chamber and the posterior chamber, which I understand is kind of confusing. Okay, and then that posterior segment is filled with vitreous humor. Whatever's behind the lens is filled with gel-like vitreous humor, while the anterior segment, both chambers, filled with aqueous humor. Essentially, the secretion produced by the ciliary, through the ciliary capillaries, okay, in the ciliary muscles. And we didn't talk about layers of the eye yet, and it's time to start with fibrous layer. Eye is like an onion, okay? Multiple layers wrapping one around another. Fibrous layer is the outermost one in the eye. There are two parts. All eye layers have several parts. Fibrous layer is not an exception. It contains two parts, the sclera and the cornea. Sclera is what you see as the white of an eye. It's the fibrous tissue, very tough, very mechanically resilient, okay? It prevents eye from collapsing under the pressure of extrinsic eye muscles, and it anchors them. All extrinsic eye muscles are attached to the sclera. Interestingly enough, sclera, if you will trace sclera, it will form the epineurium of the optic nerve and will essentially be continuous with the dura mater of the brain. It is the same type of tissue that comprises dura mater of the brain. It's pretty tough connective tissue. Does that make sense? Essentially, the main structure of the sclera is to main function of the sclera, is to provide structural support to the eye, attach extrinsic eye muscles. Have a cornea. The anterior portion of the fibrous layer is one of the eye parts that actually participates in the light refraction and responsible for us, for our ability to see things as they are. Okay? Now, cornea is bulging. Those of you who wear glasses can also see that the lens in your glasses, each lens is not flat, it's bulging. Well, depends on your vision, but I presume most of you are nearsighted, so the lens is going to be bulging. The lens are going to be convex. Sorry, no. Most of you nearsighted, so in this case, the lens is going to be concave. If you are farsighted, then they're going to be convex. Okay? So in your grandparents that cannot read the newspaper without glasses, look at the glasses, you're going to have this shape. Okay? So the same goes for cornea. If you would look at it, it's bulging here, which means it refracts the light and brings the light rays sort of to the center. It participates in focusing the light in the back of an eye. Does that make sense? Okay? Now on the outside, cornea is covered with epithelium. It's stratified squamous epithelium. And if you think about this, it makes perfect sense because your cornea constantly experiences pounding from the environment. You know, air, I don't know, wind, dust, anything, okay? So it constantly sheds the outer layer of that stratified squamous epithelium. To the opposite on the inside, the cornea covered with simple squamous epithelium, which function is what? Do you remember? What's the function of simple squamous epithelium? Simple squamous. One line of very thin cells. Epithelium is the barrier, but think about this. One line of very thin cells. Can it really protect mechanically? No. Now, is that really much of a barrier? No. So which process requires sort of a barrier, but not much of a barrier? Or transport, okay? So in this case, filtration will be mostly cuboidal cells because filtration requires a lot of ATP production and a lot of active pumping, and you know, we will see that. But yes, a lot of transport goes through the stratified squamous epithelium, simple squamous epithelium. Essentially, in this case, it's going to be pumping of sodium, which pumps the sodium out of the cornea. And this is one concept that I want to introduce to you, which will be very useful for you throughout the whole course of anatomy and physiology. Have you heard about osmosis? Osmosis. Can anyone explain what osmosis is? It's kind of a next level challenge, right? Okay, what moves in osmosis? Water. From where to where? Yeah, can you be more specific? High to low of what? Like high mountain to low mountain, what? High concentration of what? Okay, water moves from high concentration of water to low concentration of water. You're absolutely right. Do we really operate in terms of water concentration? Not really. We don't say, you know, I just bought a bottle of vodka, it has water concentration of 60%. Right? We don't say that. We usually say, you know, the solute of interest. So if water moves from high concentration of water to low concentration of water, can you say the same idea, but now in concentrations of solutes? No. What I'm saying is, look, water moves from high concentration of water to low concentration of water. There you go. Not solvents, solutes. Solutes, yes. So because high concentration of water means that there's not really a lot dissolved in it. Low concentration of water means you have a lot of solutes. So in osmosis, water goes to higher concentration of solutes. I usually give an idea, sort of like easier mnemonic rule to remember that. Water always tries to dilute. Does that make sense? If something's more concentrated, that's where water goes. Why it's so important? If sodium is pumped out of the cornea, it automatically increases sodium concentration outside of the cornea. And water will immediately follow sodium. And that rule, general rule that water follows sodium, is incredibly important in every aspect of human physiology. Whether you talk about, well, maybe not bones. Yeah. But gastrointestinal, yes. Renal, absolutely. Cardiovascular, sure. Everywhere, every place where you're dealing with osmosis, with ion transport, water follows sodium rule is just the basic. Okay, so water follows sodium, and that prevents swelling of the cornea. Does that make sense? A cornea is incredibly sensitive. Okay, if you ever touch the your eye, you're gonna blink right away. It's a reflex, because that's a normal response. When something gets on the cornea that is potentially damaging, the immediate response from the cornea is to blink and remove that, or at least deliver it to the bulbar conjunctiva, to the conjunctival sac, where tears can wash it away to the corner of the eye, to the medial commissure. Does that make sense? That's why, actually, if something gets in your eye, it may not be a most brilliant idea to immediately start rubbing it. Maybe better to just start blinking very, very fast and put some water in the lateral commissure so it flows in the natural, real way. Of course, it is not vascularized. Why? There's no blood vessels in the cornea. Yeah, you won't be able to see it, so you don't see blood vessels. Well, if there are no blood vessels, how does it receive all the nutrients and oxygen? What delivers nutrients and oxygen to the cornea? Look at the picture in the right bottom corner. Aqueous humor. Very good. Aqueous humor delivers it. Does that make sense? Another important implication of the fact that cornea is not vascularized is that we can do transplant. What is the main problem with any sort of transplantation? Rejection. Why is there a rejection? Say again. Look, I just ate, in the morning I just ate the foreign substance oatmeal, which has nothing to do with my body. I'm fine. I'm not vomiting, at least, I'm telling you, I didn't on the way here. It's not only about foreign substance. If your immune system is exposed to foreign antigens, it starts to produce immune responses. Not just foreign substance. Be more. The idea is right. The idea is right, but you have to be more precise when we talk about ANP. The thing is, your immune system, if it spots foreign object, whether it's a pathogen or it's a tissue from another person, if this person is not your identical twin, your immune system will recognize this tissue as foreign tissue, which requires immediate response and immediate destruction. That creates the problem with the rejection. How immune cells are delivered everywhere in the body. Through what? Through blood. If there is no blood vessel coming through the organ, we're good. You can easily transplant things. Does that make sense? So corneal transplantation is common and fairly easy and fairly uncomplicated by the side effects. Does that make sense? Now the next layer that we're going to talk about is the vascular layer, which consists of three parts: choroid, ciliary body, and iris. So this is choroid part. This is ciliary body, and this is iris. So choroid is the brown membrane, which is very rich in blood vessels. It's brown not because of the blood vessels, it's brown because of the pigment that's in there, and we'll talk about that pigment. Here's the idea. The blood vessels in the choroid, they supply oxygen and nutrients to all other layers in the eye, in the eyeball. Does that make sense? Now, not all the structures, but most of them. 5/6, so it's going to be what, like 84%, okay? That's about how much of an eye is supplied with the blood through choroid. You don't have to know the numbers, I don't really care, but you have to understand that choroid supplies most of the posterior aspect of the eye with the blood. The brown pigment, what's the role for brown pigment? Here's the thing, when eye is filled with the vitreous humor mostly, because posterior segment is much bigger. That's your posterior segment. When light enters the eye, what color is sclera? Sclera. Come on, look in my eye. What color is sclera? White. So when light enters the eye, if it hits the white sclera eventually, it will start to reflect from it. And it will be reflecting all over the place, right? From one corner of the eye to another. It will be very confusing. Just imagine that. That would be like you have a tiny system of mirrors in your eye that just light goes everywhere. So to prevent that scattering of light and unwanted reflection of light, this brown pigment in choroid absorbs light. So there's no scattering. Does that make sense? Ciliary body, which choroid becomes. Ciliary body is at the boundary between the anterior and posterior segments of an eye. Ciliary body consists of several structures. First and foremost, ciliary muscles. Okay, let me erase this and show you the muscles. So you can see ciliary muscles here. They are attached to the lens by the tiny small ligaments called ciliary zonules. Ciliary muscles can pull on the ciliary zonules. Does that make sense? Can you see that? Ciliary muscles, in some instances, can pull on the ciliary zonules, or should I say, they can change the tension in those ligaments. Muscles, these muscles, by moving either way, can change the tension of the ligaments. Does that make sense? Now, a little bit of a paradoxical fact, and I will repeat it several times throughout our discussion. Interestingly enough, when ciliary muscles contract, they move closer to each other. Initially, it sounds very counterintuitive. But think about this. Ciliary muscles, what shape do they have? Huh? Oh, remember, you're looking at the cross section. So what shape do they have, first and foremost? They're circular, right? They're circular. You just look at the cross section. Does that make sense? So what you see is this. Okay? They're circular. So when they contract, they become smaller. They become narrower. Okay, when they contract, ciliary muscles, they become narrower. As they become narrower, the diameter of ciliary muscle decreases. What happens to the tension on the ciliary zonules? What do you think? That's a ciliary zonule. Okay? It's like tense. My right hand up here is the ciliary muscle. My left hand is lens. Right? Ciliary muscles contract and come closer to the lens. What happens to the tension? Huh? Decreases. Does that make sense? As they relax and become wider, what happens to the tension? Increases. I spend so much time on this very small detail because it's really important for our understanding how focusing happens. That makes sense. And another reason, it's a little bit counterintuitive. You would expect, when muscles contract, they would pull on the ligaments. In this case, it's an opposite. When muscles contract, ligaments become relaxed. And when muscles relax, ligaments become tense. That makes sense. Another function of ciliary muscles, or should I say ciliary processes in the muscles, is to produce aqueous humor. Okay? Those are ciliary capillaries in the processes that produce that fluid that fills up the anterior segment of an eye. Does that make sense? So you have ciliary body, which consists of the muscle, ciliary muscle, that regulates the tension of ciliary zonules, and ciliary processes that produce aqueous humor. Now the third part of the vascular layer is iris, which consists of two muscles, circular muscles again. Practically everything in the eye, almost everything in the eye, is in the shape of a circle. Okay. The outer muscle, which has radial muscle fibers, is called dilator pupillae. The inner circle, which has muscle fibers in a concentric shape, is called sphincter pupillae. Both sphincter and dilator pupillae regulate the diameter of the pupil. Make sense so far? You follow me? So think about this. When sphincter pupillae is stimulated, then muscle fibers contract, making the pupil smaller. When dilator pupillae is stimulated, muscle fibers contract as well, making pupil larger. Does that make sense? Constriction of iris and constriction of pupil is under parasympathetic control. So, for instance, right now it's hard to say because right now you have two stimuli that are kind of opposite. One stimulus is the bright light. So your pupils are really narrow. On the other hand, you have to, look farther than your far point of vision, so it contributes a little bit to the dilation. You have to understand it's not like on-off. It's not like one or another. They both respond. Does that make sense? Like if you try to read in a dim light, your pupils will dilate because of the dim light, but they will partially constrict because of reading. So there's going to be some sort of the equilibrium in between dilation and constriction of the pupil. Does that make sense? But of course, when the function of your oculomotor nerve is tested and the doctor shines the light into your pupil, there's no reading component or looking component involved. That bright light stimulus overruns everything else. Does that make sense? What else can stimulate dilation? Dilation is stimulated by any sort of excitement, whether it is positive or negative excitement. Well, by negative excitement, I mean fear. By positive excitement, I mean speaking to someone who's appealing to you. Right? Constriction, on the other hand, can be either stimulated by relaxation and reading or speaking to someone who's repulsive to you. Which makes sense, because if you talk to someone repulsive, you want to see as little as possible of this person, so you want to constrict your pupils. A joke. Now, the color of the iris, what determines it? The same brown pigment, melanin, that can be found in choroid, determines the color of the iris. So when we say people have different eye color, it's all same melanin. It's only the amount of melanin that matters. Does that make sense? So say I have brown eyes, somebody else has gray eyes. We all you have gray eyes, right? Blue, whatever. A lot of it is just color perception. But you have less melanin in your eyes than I have. Does that make sense? Also, kids, newborns, often have gray eyes, and later in life, they become brown. What happens? Say again? Yeah, melanin production increases. And that actually, I try to kind of tie this to the concept of genetics. Think about this. What is melanin? Chemically. Which biological macromolecule that is? There are not so many: carbohydrates, proteins, lipids. So melanin, what do you think is? Most likely. Traits in people are determined mostly by, starts with P. Proteins, yes. Melanin is the protein. Proteins are encoded in what? DNA. In the DNA. In the genes. So it means that when baby grows, during those developmental stages, the expression of the gene that controls, that produces, that responsible for melanin production increases. That's it. Does that make sense? We talked actually about this when we talked about skin color, and we talked that people with vitiligo or albinism, especially albinism, okay, they do have a gene for melanin. They just don't have this gene expressed. There's no production. They have the gene. Does that make sense? The next layer that we're going to talk about is the retinal layer. So that's the innermost layer of an eye. By innermost, I mean that this is the retinal layer. Do you see what I'm saying? It's closest to the eye, to the center of an eye. And structurally, it is the extension of the brain, which makes perfect sense. Sclera is extension of dura mater, and retina is extension of the brain. What does brain consist of? Which cells? Functionally. A lot of glia, we understand that, but what's neurons? Neurons. So retinal layer, to a large extent, are neurons. Even photoreceptor cells that you may have heard about as rods or cones are essentially slightly transformed neurons. Does that make sense? Of course, there are some glial cells, as there are in the brain. Those glial cells form pigmented layer. Here in this picture on the right, you can see pigmented layer barely. Here, as between, deep to choroid. So this brown part is choroid, and deep to it, immediately deep to it, is pigmented layer. It's a single cell thick layer. Those pigmented layer, first of all, participates in light absorption to prevent scattering, but what's most important, it's sort of a recycling plant. Those neurons that receive the light, that process the light, the photoreceptor cells, since they are exposed to the light, they are getting damaged by the light. And the fragments, they're getting damaged. They're fragments of the membrane that kind of fall off, and parts of the cell that die. Pigmented layer, cells of pigmented layer, phagocytose those fragments and recycle them. Does that make sense? That's surprisingly efficient. They also store vitamin A, which is necessary as the precursor for photopigment in your photoreceptor cells. So that's pigmented layer. Think about pigmented layer as the helpers, okay? Sort of a team that supports the stars, the neural layer. Neural layer consists of 3 different types of cells: photoreceptor cells, bipolar cells, and ganglion cells. Photoreceptor cells surprisingly receive the light and convert it into the nervous signal. Bipolar cells convey that signal to ganglion cells, and axons of ganglion cells then form optic nerve. Okay? Can you see that? We can try and, do some kind of a picture here. Look at this. If this is pigmented layer, these are photoreceptor cells. These are bipolar cells. And these are ganglion cells. Does that make sense? Can you see that? And then axon of ganglion cell kind of rolls around and forms optic nerve. Okay. Now my quick question, first of all, each of these cells, sorry for the picturing, right there, that this red and, so photoreceptor, bipolar, and ganglion. They're all neurons. What type of neurons are they structurally? Are they multipolar, bipolar, or unipolar? The answer is kind of written right there. Bipolar. They're all bipolar. Okay. That's one. Second, what is this? What's here? Well, those are synapses. That makes sense? Now, if that's synapse, for instance, what is that? And here's the information flow. It goes to the brain. What is that? This. Okay, that's a neuron. Huh? Say again? Huh? Say again? Nope. It's the neuron. Well, not really. No, it's like a relay. Okay, we will talk about the function of bipolar cell. What I mean is this part of the cell. What is that? It's not an axon. If it's not an axon, then it's no. I think I'm pretty positive, pretty good with depicting body. Dendrite. That's a dendrite. Does that make sense? Dendrite, axon. Dendrite, axon. What do you expect from me? I still have no idea how I got B for art. Okay? Does that make sense? Think about this. Now this is sort of I want this this concepts to start clicking in to start linking together. What is the function of dendrite? To receive the signal. What signal does the photoreceptor cell receive? Light. This dendrite will receive light. What's the function of axon? To transmit. So you always have to think. You identify the flow of information. And think about this. Information always goes from retina to the brain. Right? Because it's 100% sensory nerve. There's no other option. There's nothing to send to retina. There are no muscles there. There's no response there, no glands, nothing, no effectors. Does that make sense? So you identify that flow and then say, okay, which part of the cell is on the end of that flow? It's going to be axon, right? So which axon is in the end? Here. That's axon. That's incredibly long axon of the ganglion cell. That's all thing. This whole thing is the axon. Now, if this is axon, that must be a dendrite. Now, if that's the dendrite, that must be an axon too, because you never have synapses between dendrites. You can get, axon to dendrite, axon to soma, yeah, but never dendrite to dendrite. Do I make sense? And you have to understand that the whole idea of information flow from retina, it's like this. That black line, black arrow, shows you how information goes from retina into the optic nerve. Does that make sense? Awesome. Now, when optic nerve leaves the retina, here's a little problem. That place, nerve leaves, there's no rods or no cones, no photoreceptor cells here. This place is called blind spot. If light gets on this place, it is not sensed. Try to analyze the picture that you see now. Do you have a tiny little black dot in the center of the picture? Like whatever you look at. It doesn't matter. Like, do you have? No, you shouldn't. I mean, if you have, then you should go to the doctor. You don't. Why? Not the eye, the brain. It's pretty much, look, it's actually fun sometimes to try to focus on your body activities. Sort of activities that you don't really focus on in regular life. You know, how you walk, how your eyes move. And you will notice that your eyes constantly moving. Even if you, like, they jerking a little bit. And as they jerk, you have multiple pictures overlapping. And that overlap compensates for the blind spot. Does that make sense? So it's the conscious feel of the blind spot. Does that make sense? Okay? Now, photoreceptor cells. It's sort of an enlargement of what we saw. Sorry about that. So let's take a glance at the structure of the retinal layer in a little bit more details. As I mentioned, there's a tiny one cell thick layer of pigmented cells. Okay? Then you have photoreceptor cells. Then you have bipolar cells. And then you have ganglion cells. Does that make sense? Okay, that's on the right, it's microscopical image. So that's what it sort of looks like in the real life. This is the artist's depiction on the left of rods and cones. Rods and cones. So you can see that rods are first more numerous, right? And they shaped like rods, the outer segments. shaped like rods. Outer segments of cones are surprisingly shaped by cones, like cones. Rods and cones, they all communicate with these pink bipolar cells, and bipolar cells then communicate with ganglion cells. Does that make sense? And then ganglion cells extend their axons, forming the optic nerve. Does that make sense? So rods are specific for dim light. Rods are used when you look at something in the dark. Also, they are specified for the peripheral vision. Essentially, think about this. It's actually a great picture here. Let's trace a few rod cells. Okay. Let's take a look. What would be the best? Okay, that's fine. Can you see these three rods right here that I'm circling now? They all converge in one bipolar cell, and that bipolar cell transmits the signal to this ganglion cell. Does that make sense? So sensor input from three rods is transmitted to a single ganglion cell. It's like amplification. Think about this. One thing is when you get one punch from one person. Another thing when three people simultaneously punch you in the face. That signal is going to be much harder. Does that make sense? Much more sensitive. They have higher sensitivity. On the other hand, think about this. Somebody punches me in the face. I can definitely say where this punch is coming from. If three people punch me in the face all at the same moment, it's really hard for me to distinguish between the punches. It feels like a giant punch, very kind of diffuse one. So same happens with rods. Think about this. Do you see the your environment now focused and colored and bright, right? Now imagine we turn off the light completely, just obliterate the light in the hallway everywhere, and it becomes dark. Maybe not pitch dark, but very dark. How is it going to change your perception? No colors. What about details? Okay, no clear, no sharp, because rods will take over. Does that make sense? Sensitivity will increase, but at the expense of sharpness and detailization. Does that make sense? Now, cones usually have, well, not usually, each cone has its individual pathway to the brain. Literally, each cone sends its own unique signal to the thalamus. Does that make sense? Essentially, sometimes I compare it to the one singer versus the choir. If the person sings to you, you can usually hear all the words, you know, and it's really sharp, and you can tell what the song is about. But if you will step away from that person far enough and person doesn't use the microphone, you will stop hearing this person. The signal is weak, but very clear. Now, if you have like 200 people singing the same song, they're going to be awfully loud. But the precision, the sharpness will be not comparable. Does that make sense? So that's the fundamental difference between rods and cones. Rods are very sensitive, but do not provide good resolution or color. Cones are not sensitive. Okay, the limit of activation is high, the threshold is high, but they provide very sharp vision and colored vision. Does that make sense? Did I want to say something? No. No, I don't think so. Well, now, macula lutea. You may have heard about it in the context of macular degeneration. Macula lutea, the literal translation of this word, macula lutea, is the yellow spot. And guess why it is called yellow spot? Because if you do, what's the word for it? The examination, fundal examination. You know, when the doctor looks at the bottom of your eye, that's called fundal exam. When doctor does fundal exam of your eye, macula lutea looks like white spot. Okay? That's the area which has the higher concentration of cones. That's the area where the image is focused. Does that make sense? The highest concentration of cones in the macula lutea is the area that I circled, very tiny area called FOVEA or FOVEA centralis. FOVEA centralis, that is very small, 0.4 millimeters. The penny is about 1 millimeter, maybe a little more than 1 millimeter. So the diameter of FOVEA centralis is less than the thickness of a penny. And FOVEA centralis contains only cones. Only cones. Everything that is important, like you look at the image here, you say you focus on FOVEA centralis, that FOVEA centralis is focused on your FOVEA centralis. Does that make sense? Everything important is focused on the FOVEA centralis. And then the number, the percentage of cones progressively decreases as you move from FOVEA to the edges of the macula lutea. Macula lutea, potentially its yellow color may be determined the pigments lutein and zeaxanthin, which may serve as a natural sunblocks. Think about this. There's enormous amount of light, sunlight, that gets in your eye, including UV light. UV light is really damaging to the cells, so those pigments may serve as the sunblocks. Okay. Now, when you drive, you play sports, anything where the environment changes, okay, and this happens, your eyes jerk all the time, so that image can be, whatever is important, can be immediately focused on FOVEA. Even like in the regular, if just environment, just, you drive down the road, you may just go straight, cruise control, you don't pass anything like this happens, but still your eyes are jerking all the time. So you have an exact sharp perception of the environment around you. So lens, that's the bulging structure that's most importantly flexible. Lenses are biconvex, so they shape like this. They are biconvex and they can change the thickness. They don't change shape, so lens, look at this. Your lens are not going to be like this. They cannot do that. But what they can do is this. They can flatten and they can bulge. Do I make sense? Now, let's go back for a second to the ciliary body. One of the components of the ciliary body is ciliary muscle. Remember that circular muscle? Which ligaments are attached? The zonules, ciliary zonules, right here. When the muscle relaxes, what happens to the zonules? When the muscle relaxes, they get tense, right? As they get tense, what happens to the lens? They get flat. Does that make sense? because ciliary zonules are attached to the ends of the lens. As the zonules are pulled to the sides, lens become flattened. Does that make sense? Okay? When the muscle contracts, what happens to the zonules? They relax. Remember, lenses are elastic. What happens to the lens? They start to bulge. What changes as the lens flatten or bulge? What component of vision? Your focus, yes. And as they become bulged, do you see close? I mean, is it for close vision or far vision? Look at the, if, like, my mom cannot read anything close. So if I would look at her lens in her glasses, it would be like this. Because she needs to bring the image closer to her. Does that make sense? Okay? So convex lens, when they bulge, they allow you to see something up close. That makes sense? Okay. One more thing. When they flat, obviously it's for farther vision, right? So if I want to look at that schedule that there in the door, my lens is flattened. I don't see a thing anyway, but I can see the schedule. Okay? Does it require any effort from me? Do I get tired of looking at the schedule? Well, mentally I am, yeah, but not physically. There is no tiredness because my ciliary muscles are what? When I look far, relaxed. If I start to read something, my ciliary muscles do what? To make lens bulge. They have to contract, okay? Lens bulge. They get tired of reading. Physically, yes. Ciliary muscles get tired of reading. Does that make sense? That's why your eyes get tired when you look at something up close. Does that make sense? Now, so lenses obviously are avascular as well. Otherwise, we would be able to see those blood vessels, right? I'm going to ask you again, how do they receive nutrients and oxygen? Through what? Aqueous humor, right? They consist of so-called lens epithelium, the cuboidal cells. Cuboidal cells, when they become differentiated, they turn into fibers that enable those lenses to become, elastic. Okay. Those fibers have a lot of protein called crystalline. Fibers are essentially dead cells, but crystallines, they form the body of the lens. And as we grow up and we become older, more and more fibers are added up. And cells become more bulky. Okay? They become less elastic because old crystalline proteins maybe denature or oxidize, so that it impairs the elasticity of the lens. And we lose the visual acuity. We cannot focus as well. Does that make sense? Now, all of those structures, retina, well, choroid, right? Choroid supplies blood to the sclera, but where this choroid receives the blood from? So there are two blood vessels, major blood vessels, central artery and central vein. Okay. Central artery and central vein, they bring blood to the retina, to the inner two-thirds. Essentially, they bring blood to the neural layer of the retina. Most of it. And if there is any sort of vascular pathology, any sort of vascular disease, it will first of all affect eye, and second will be seen in the eye. Do I make sense? Will affect the eye and will be seen in the eye. For instance, hypertension causes structural changes in the vasculature, structural changes in the blood vessels. So during fundal exam, those structural changes in those blood vessels can be seen, and the person can be diagnosed with hypertension or pre-hypertension. Now, look, hypertension itself is bad, but it's not hypertension that kills people. It's the vascular changes, vascular remodeling that kills people. And I'm kind of getting in my soapbox here. I was studying microvasculature, small vessels, for 5 years, and I can tell you that the first signs of vascular disease are observed in the small vessels. Your large arteries may be in perfect shape, but first signs of disease can be noticed in much smaller vessels, much earlier. So when doctor, when optometrist or ophthalmologist looks at your blood vessels, that person, that doctor can spot structural changes in those tiny blood vessels and tell you, look, something is going on with your vasculature. You better check. Maybe it's, pre-diabetic state, or maybe your hypertension is, at the level too high. You really have to focus on that. You really have to go to cardiologist. Does that make sense? So it has enormous diagnostic value. Another interesting pathology that's associated with the blood vessels in the retina is called diabetic retinopathy. What's diabetes? Come on. Say again? Too much sugar in the blood system. What else? Most important. Huh? Say again? Okay, what else? Come on, people. It's the staples of American culture. Diabetes is the foundation of American culture, along with Mickey Mouse and muscle cars. Diabetes has maybe of two types. Type 1, which is genetic. You cannot do anything about it. And type 2, which is acquired, usually manifests at the age of 30, 40, and associated with a lifestyle. However, there is a genetic component to it. Don't get me wrong, there is a genetic component, but it's mainly lifestyle disease. Both conditions are characterized by elevated levels of sugar in the blood, which by itself, I mean, if you think about this, so what? Vampires would probably have it like a dessert, right? But it turns out that when there is an elevated level of glucose in the blood, glucose gets oxidized, forming, and glucose also modifies proteins and cell walls, forming so-called advanced glycation products, which are, to say the least, bad. Okay, essentially, high glucose in the blood causes inflammation of the blood vessels. So far, do you follow me? Now, inflammation, is that a normal response? Yes, it's normal response to what? To injury. Now, when you have just high blood sugar and your blood vessels start to get inflamed, is there any real injury? No, but you still get inflammation, right? And all consequences associated with it. So what happens, bless you? So what happens in the diabetic retinopathy? Elevated blood sugar causes inflammation of the walls of capillaries, and walls of capillaries are really thin. It's only endothelial cells which get inflamed, surrounded by pericytes, which support the structure of endothelial cells and endothelial, the vessel wall. Okay, when pericytes become dysfunctional, okay, the junctions between endothelial cells form tight go to wide open. And that leads to the spillage of plasma out of the blood vessel. Does that make sense? So essentially, I have leakage of plasma from these tiny capillaries into the retina. With plasma goes sometimes red blood cells. So you have migration of inflammatory molecules, and you have red blood cells in the retina, and they're not supposed to be there. So retina gets inflamed. Does that make sense? It's sort of, kind of alright. It's non-proliferative, which means nothing tries to compensate for it yet. Now think about this. All those capillaries are essentially little tiny pipes that supply blood to the retina. If pipes become leaky, what happens to the retinal blood supply? It decreases. How can you compensate for the decreased blood supply? How can you compensate? You have a bunch of pipes that are leaky. You can't. It's too local. It's a good idea, but you can't. Fortunately, you can't. Can you plug them? No, because they inflamed and they leaky already. What is the only thing you can do? Well, the eye will die. The eye will die. It's not an option. You still have blood supply. How can you bring it back to normal? Can you build more pipes? You sure can. Okay? So new capillaries will start to grow. Are they normal? No, they're also leaky. So in proliferative diabetic retinopathy, you have a bunch of new capillaries that will grow. All those capillaries will be leaky. So it will be positive feedback. Okay? Worsens, the blood supply worsens. It leads to more capillaries, which leak more plasma in the retina, which gets more inflamed, requires more blood supply, which leads to the growth of more capillaries, and eventually it leads to the macular edema with a bunch of hemorrhages, and if anywhere there is inflammation it resolves with fibrosis. Obviously, fibrotic tissue has nothing to do with the normal retinal tissue, so eventually it leads to the loss of vision. It's a leading cause of blindness among the adults of working age in the world. Now here's the question. I hope the picture that I pictured here is pretty grim. It's pretty bad, right? Can you prevent that? Yes. Well, some people don't have that option. Type 1. They don't have that option. Can you still prevent that? The answer will be as simple as it is boring. You can by regularly going to your doctor. That is it. Because first of all, there are drugs to treat diabetic retinopathy. Second, if the person properly maintains the blood levels of the blood sugar, okay, checks for the integrity of the retinal circulation, diabetic retinopathy may be delayed by decades. Instead of developing it at the age of 40, it may start to develop at the age of 60, 70. And that's a great example of, preventative measures. Just being, compliant, going to a physician and getting checked regularly will prevent the development of diabetic retinopathy. Essentially will leave you, with your vision. It's great, to see things. Now, since we are sort of in a dessert mode, like the tastiest stuff, the diseases and things. Another disease is pupilledema. That's actually a very interesting disease. What happens, your brain is surrounded by the liquid. How do we call this liquid? No, your brain. Cerebrospinal fluid. And your eye is to your brain what? Is your eye anatomically a part of your brain? I told you in the beginning. Yes, it is. It's the extension. It's the extension. Nerve, optic nerve is the extension of the brain. Sclera is the extension of dura mater. Does that make sense? So, when the intracranial pressure, the pressure in cerebrospinal fluid increases, that fluid is being pushed down through the optic nerve. It increases the pressure on the optic nerve. Optic nerve first engorges, swells, and as it is being pushed into the through the optic disc, through that blind spot, it is being pushed inside the eyeball. Does that make sense? You can see it progresses, that's normal optic disc, and then optic disc becomes larger and more blurred. Okay? Does that make sense? As it being pushed farther and farther inside of the eyeball. Actually, at a certain point, there's a protrusion of optic nerve inside of the eyeball. And that protrusion, essentially, when neurons that are in the optic nerve are swollen, there's a compression in them, they are dysfunctional. So there's no proper signal transmission, it may lead to blindness. Okay? And interestingly enough, there's a group of people for whom papilledema is sort of a professional disease. People who experience changes in the pressure, environmental pressure, on the regular basis, sometimes several times a day. Huh? Pilots. Yes. For pilots, papilledema is the professional disease. A lot of studies of papilledema is done on pilots. Okay? There is technique, pretty much no prevention, just, don't expose people to regular changes in the blood pressure and intracranial pressure on a regular basis. Does that make sense? Not so much age related, it's more occupation related. Unless there's, now think about this, if there's someone has hydrocephaly, it's an increased intracranial pressure immediately, and it may immediately lead to papilledema, which will not be the only concern of the person with hydrocephaly. It will be just one of the manifestations. Does that make sense? Now, another condition that is pretty well known is the retinal detachment. You can see it right here. Detached retina. Usually it's a result of a blow. When pigmented layer and neural layer separate, the vitreous humor gets between them. Neural layer has no blood supply anymore, and neurons that cannot survive without normal blood supply start to die. Okay? If retinal detachment is spotted quickly, it can be repaired. The laser kind of reattaches it back. Okay? But if not, it may lead to the loss of vision. Very common in boxers due to the blow to the head. Couple more conditions that you may have heard about: glaucoma and cataract. Glaucoma, as we discussed, is the condition associated with an impaired drainage of the aqueous humor. So that's normal eye. Aqueous humor is produced in the ciliary, by the ciliary capillaries in the ciliary processes, and it drains through the posterior chamber, through the pupil, into the anterior chamber. And then it flows into the scleral venous sinuses, here, the corneoscleral junction, and goes back in the blood. So you have normal flow of aqueous humor. If for some reason scleral venous sinuses are blocked, fluid cannot get back into the blood, and starts to accumulate in the anterior segment, and it exerts additional pressure on the posterior segment. Posterior segment transmits that pressure on the retina and the optic nerve, and that pressure on the neurons in the retinal layer impairs their function. So this is how it goes from the normal vision to the glaucoma. You see that the field of vision progressively decreases because of the degradation of neurons in the retina. Glaucoma can be treated, okay? Sometimes pilocarpine is sufficient. Usually the treatment is the increase of the drainage, dilation of the scleral venous sinuses. It's a drug called pilocarpine. It's actually parasympathomimetic. Dilates those vessels. In some cases, laser therapy is required. Now, as far as I know, never had any treatment like that, but as far as I know, laser therapy is pretty common and it's not extremely complicated. So it can be done, and glaucoma can be treated like for life, and you won't need a lot of drug interventions. Okay? Another condition, cataract, is often congenital. Congenital cataract is cloudiness of the lens when crystallines are being oxidized. Well, it is one of the proposed mechanisms that increased oxidative stress. Do you understand the concept of oxidative stress? No? Good, because I hate this concept. It's like a perfect thing, like, you know, what is responsible for this disease? Oh, it's oxidative stress, which doesn't explain anything. Like, our cells are under constant oxidative stress during cellular respiration, and we don't die. The idea is that, idea of oxidative stress is that in the human body, in every cell, we do produce a lot of so-called reactive oxygen species. It's a little bit of digression. I'm not going to ask about ROS, reactive oxygen species, on the exam, but it's a concept that you should know, okay? Reactive oxygen species. Two most notable reactive oxygen species are superoxide anion and hydrogen peroxide. Both of them are normally produced in the human cells. Both of them are very oxidative and chemically active and can destroy a bunch of intracellular structures and modify molecules. So far, do that make sense? Humans have enzymatic systems that deal with reactive oxygen species and neutralize them. So hypothesis about reactive oxygen species or oxidative stress is that if those compounds are produced in extremely large amounts, enzymatic systems in our cells cannot deal with them. Think about this as the garbage in garbage trucks. If everybody will start to produce a full can of garbage every day, but we still have garbage trucks running once a week, our streets will become filthy very quick. Does that make sense? So the idea is that oxidative stress, that overproduction of molecules of oxidative stress, which are, by the way, associated with inflammation, can damage cells, like cells of the blood vessels, cells of the muscle, whatever cells they are in. Does that make sense? Including lens. Can we prove the oxidative stress theory? The answer is no. There are experiments that suggest, mostly on mice, that if you will start giving mice some kind of antioxidants, like vitamin E, for instance, vitamin D, it often decreases the risks of developing, cardiovascular disease. or, I don't know, diabetes or something like that. Does that make sense? So far, from what I told you, does this idea of oxidative stress, does it look legitimate? I mean, yeah, I mean, there is an ample experimental evidence that antioxidants can prevent or decrease the symptoms of cardiovascular disease, at least in animals. Here's a little problem. You should always ask, okay, human cells produce those chemicals, oxygen, superoxide anion and hydrogen peroxide. What for? And the answer is, they are maybe anti-cancer chemicals. It was shown that in some cases, cancer cells, lung cancer cells, can be killed by those chemicals that are produced in those cells. So potentially that oxidative stress is the mechanism to destroy cancer from the inside. And there was a study that made quite a big splash when mice, the model mice that developed lung cancer, were treated with antioxidants, drugs that prevented oxidative stress. Guess what? Lung cancer in those mice developed much faster and more frequently. That kind of threw a huge stick into the wheels of that oxidative stress train, you know. So now people kind of reconsider, do we really need to take all those, you know, supplements and antioxidants and like, oh, you gotta drink like pomegranate juice because it's so good for your health and stuff? And I don't have an answer. I do not, but kind of try to think. So there's an idea. Oxidative stress and metabolic alterations. Of course, smoking is great, you know. Not smoking is great. Smoking is the great, you know, answer to all the problems. Why this person has heart disease? Oh, smoking, you know. This, smoking, that, smoking, cataract, smoking. It was really associated with smoking. The risk for developing cataract. But if it's oxidative stress, then consumption of such well-known antioxidant as vitamin C should prevent that. Well, there's multiple studies that show consumption of vitamin C is associated with increased risk of cataract. So it's not that clear. But to the treatment. Say again? No, well, to consume that much vitamin C on the regular basis, you really just have to take supplements. You cannot get it from the food. There's no way. I mean, you probably have to drink like 3 gallons of orange juice a day. So, not possible. But there's a treatment to cataract. You can just replace the lens. They're artificial lens and they work fine. It's surgery, but it can be done. Okay. Any questions? Okay, here's what we're going to do. We're going to take a break, and then we're going to try to work with those models. Some of them, they're a little outdated, but we'll try to carefully look at them, look at the muscles, and we'll try to tear them apart. Just a little bit. Okay?